Okay, so I think if we're all getting settled, we'll just have everyone take their seats. And I want to welcome everyone to the second part of our session today. It's been a great morning so far. I know I've learned a lot and really feel like it's a privilege that we get to share in this room so many interesting ideas and recent findings. And so it's really an honor for me to be standing here to present to you today Dr. Patrick Brunner, who is an IEC counselor and an associate professor in dermatology and director of the CTCL clinic, the Cutaneous T-cell Lymphoma Clinic, at the Kennedy and Eric J. Waldman's Department of Dermatology at the Icahn School of Medicine at Mount Sinai in New York City. And as a clinician scientist, he has an interest in chronic inflammatory diseases, working with vitiligo, atopic dermatitis, alopecia areata, and in particular interested in immunobiology, not only in inflammatory diseases, but also in malignancies of the skin. Dr. Brunner obtained his degree from the Medical University of Vienna, where he trained with Dr. George Stingel, and has then moved to New York and trained with Dr. Gutman-Yaski and Dr. Kruger, and where he now is situated and bringing his research to us today. So without more ado, I'll introduce Dr. Brunner. Come on up.
Thank you so much for the kind invitation. I'm thrilled to talk about navigating big and/or metadata, which in AD, which of course affects us all, because all these many data sets and this high abundance of data sets that we are all, or that many are facing, is of course one of the major challenges in biomedical research. These are my disclosures. So you've all, I'm sure, seen such a graph that compares the amount of data that's being generated in the world. And the question is, what actually is big data? There are several definitions of it. Here you see the number of bytes on a logarithmic scale. And when you just look at what signifies a single letter or a single page or a chart, it's much lower than, for example, what our brain can store, or all the words that have been spoken by humanity. But when you look at the healthcare data in 2017, all the combined healthcare data in the year 2017 in the United States, it's a factor 1,000 higher. So all the data that's being generated every year is, of course, exacerbated more and more. And it's, of course, one of the major tasks of biomedical research to process these large data sets. When we talk about big data, there's not only the sheer volume of the data, it's also the question, how quickly is data produced, how quickly is data generated, how fast does it change, and of course the variety of the data, because ideally you have a cross-talk or a cross-analysis of photos, audios, videos, and so on with all these big data sets. Particularly in atopic dermatitis, I think many attempts to analyze the disease has been in order to bring order to the chaos, so to say. It's a heterogeneous disease. Many patients have different subtypes. We are not really there to stratify patients according for endotypes or for treatments, but nevertheless, I think many of those big data attempts or data sets that have generated big data have been attempted to better understand the pathophysiology. And it's a skin disease, of course, and this is why the analysis of skin is very important, and you're very familiar with this, with all these omics, be it genomics, transcriptomics, proteomics, or metabolomics. So all these are different levels where we can study the skin and try to better understand what's going on in the skin in order to find treatment targets and, of course, treat those patients. And all those approaches were, of course, extremely successful given all the new treatments that started with psoriasis, then continued with atopic dermatitis, and are now going into vitiligo, alopecia areata, and so on. when we talk about omics, of course, transcriptomics and proteomics are maybe the most widely studied next to genomics. Transcriptomics, because it's when you want to analyze tissue, it's often most straightforward to look at the RNA because it's relatively easy to analyze and quantify, much more straightforward than looking at all proteins, for example. I don't want to bore you too much with details on single-cell RNA sequencing. You're all aware that this is now a method that is very widely used in order to study complex tissues and has really revolutionized how we can look at individual tissues, including the skin. There are pitfalls to it, and it's not a solution for everything, of course, but when you have individual or when you have a complex tissue, when you can analyze or isolate individual cells and you can analyze the transcriptome of each cell, that's of course a major advantage compared to isolating a biopsy, processing it, and getting one data point as opposed to maybe 10,000 data points in single-cell RNA sequencing. Concerning the single-cell RNA-seq workflow, there have been, or initially there have been plate-based techniques where you just sort one cell into one vial and then analyze it, but I think now this droplet-based technique that is commercially available has now become widely used, and it's what's being used by many or by most investigators. Once you encapsulate a single cell into such a droplet, you can take the transcriptomic content with a barcode. The cells are broken down, sequenced, and then with complex bioinformatics analysis, those cells are reassembled, and those single-cell RNA-seq analysis can be performed. When you look at all these data sets, at the end, of course, you want to have a nice plot in a two-dimensional space, but there are, of course, many steps in between that are often a sort of a black box, and that's also where errors can occur. First and foremost, it's, of course, important to make sure that the barcodes, the labels, and the data that you're looking at really corresponds to living cells, that you're not counting trash. And it's also important that you have a sufficient depth. So about 1,000 genes per cell is sort of a threshold, I think, in order to conduct meaningful analysis of single-cell data. And you also, of course, need to filter out doublets or eliminate dying cells or cells with broken membranes that have then a bad quality. And there are certain ways to do it. Look at mitochondrial RNA or ribosomal RNA, and by these bioinformatics analysis, you can filter out low-quality cells and then end up with cells or with data points that are of sufficient quality. With all these big data, one or the biggest challenge remains how to transfer these big data that has multiple dimensions into a two-dimensional space that you can print on paper, because already three-dimensional space printed on paper is a challenge to see it. So you really need to reduce it into the two-dimensional space in order to capture it, so that also our brain can process it and that we can visualize it. And this happens via these methods. Initially, or for a long time, principal component analysis were sufficient to plot sort of certain data sets, but with single-cell RNA sequencing, you need more elaborate algorithms, and the two most prominent are TSNE and UMAP. UMAP is now really established that everyone does. Both are useful algorithms. UMAP has just the advantage, I mean, when you look at these plots, each dot is a single cell, and this is how you lay it out, and cells or dots that cluster closely together are more similar, and those that are farther apart are more distant, and that's how you identify all those cell types in single-cell RNA sequencing. And the advantage of the UMAP is really that it gives you a good overview of the overall landscape of your sample, whereas TSNE is more of a zoom in. But so right now, UMAP is really the one that's being used by many investigators. One problem is, of course, that when you process samples at different days, different times, or with potentially also different protocols. You get batch effects, and you need to correct for those batches. There are some algorithms out there that can do that. When you look, for example, at this data set where the color shows you different samples, you see that there seems to be some sort of artificial or some stripes here in this cluster, which most likely is derived from batch effects, and you need to then calculate or eliminate those batch effects with those algorithms in order to get an even distribution that makes most sense. And then you can look at which markers are expressed by which classes, and then you end up with a nice plot where you can identify all those cell types and potentially identify new cell types. And when you have gone through all of these processes, you, of course, you have an extremely valuable data set, because you're not looking at a single data point, you're looking at individual cells, and you can do several calculations or analysis. Of course, you can compare two groups and calculate the mean. One group might have a higher mean, the other one might have a lower mean. You can look at variability, of course, that you can't do with bulk analysis. You can see some cells might have a high expression of some gene, some might have a low expression of genes, some population might be more homogeneous than the other. And of course, you can do a so-called pseudo-time analysis where you align groups according to their expression levels from low to high or high to low, and then from that you can infer certain biological developmental stages, which of course needs to be confirmed in a functional assay. But with these methods, you can extrapolate a lot on the biology of these conditions. So transcriptomics is really, because analyzing mRNA and cDNA is most straightforward, really the most widely used method. Proteomics is more complicated, because in order to analyze and process and analyze complex protein mixtures, it requires more complicated methods, and one sort of help to better understand proteomic mixtures of tissues, of blood and so on, are multiplex assays. So multiplex assays such as Olink assays are not sort of a proteomic approach. There are panels out there, curated panels, that can analyze hundreds of different proteins within a sample. For example, this platform consists of two monoclonal antibodies that bind the target, and a hybrid and attached DNA can hybridize when those antibodies bind their target. And then it's not read out via proteomic approach, but via actually a qPCR approach, and with this you can calculate the protein abundance. So multiplex assays are much more straightforward to use and much easier to use, and with this you can get a lot of information out there and can of course calculate different protein concentrations in different samples. When we talk about big data, we of course need to think about how do we get our samples, because garbage in, garbage out. You need to make sure that you use the sample that is appropriate for the questions that you're asking. The gold standard, of course, for skin investigations are skin biopsies, but as you all know, not every patient is happy about a skin biopsy. It can superinfect, and it will for sure leave a scar. Dr. Gutman has already told you about tape strips, which is a very elegant method, a minimally invasive elegant method to capture not only proteins but also mRNA from the skin. Depending on the question that you're asking, it's a very powerful tool to monitor disease and to also quantify certain inflammatory and barrier markers that are in the skin. Another approach that we took was a so-called suction blister approach, because for single-cell RNA sequencing you need intact cells, and this is what we tried. Suction blistering has been around for a very long time. You have a negative pressure chamber that you mount on your skin that's connected to a vacuum pump, and over the course of 1 to 2 hours you get formation of these little blisters, and those little blisters are filled with interstitial fluid that you can harvest with a small needle, and the roof of the blister is the epidermis that you then can very easily cut off with little scissors and digest it quickly, and then you can perform cellular analysis with this. It's a method that is not really painful. There's a bit of a tingling, and because it's the split is the basement membrane, it doesn't go deeper. That's why it doesn't leave a scar. And when you do single-cell RNA sequencing and when you compare biopsies, for example, with blisters, you see that many cell populations are equally well distributed in both methods. You have lots of keratinocytes, you get lots of T cells, and we now know that the T cells are not only the ones that you passively or that you capture with this within the epidermis. You get, especially of migratory cells, highly migratory cells such as T cells, they actively migrate into the blister fluid, so you also will get cells from the dermis. And you get also lots of dendritic cells. What you do not capture very well are macrophages because those are much more sessile. As you know, when you get tattooed, Dr. Krüger showed that those cells are containing or characterize them as the major containers of tattoo dye, for example, and tattoos are permanent, of course, so those macrophages do not like to migrate a lot. And of course, when you want to look at endothelial cells, smooth muscle cells, or fibroblasts, you might not be able to do that. Therefore, you need a skin biopsy. Why am I showing you that? Because the method that you do often gives you or the method that you choose often has an impact on the data that you analyze in a way, how good is the quality of the data. And the problem is when you take biopsies, you can often have cell damage, and the suction blister approach seems to be less disruptive to cells when you directly compare them. How do we know that? One big issue with single-cell RNA sequencing is that you do not always only get the cells that you are analyzing, you also get or cells, when they are dying in the process, they can release their mRNA. And these especially high abundant mRNA, such as keratins, can then contaminate all your other cells. And this is called ambient mRNA contamination. And when you look at keratin 6A, for example, this is a molecule that should actually be only present in keratins. So we are pretty confident that they're only present in keratins, right? So when you look at, or when you compare an analysis from a punch biopsy with the suction blister approach, you see that there are nuanced differences. I mean, when we look at keratin 6A, we see red is upregulation, that the signal is primarily within keratinocytes here and here. But you also get this low-level information here in T cells or dendritic cells, and T cells and dendritic cells should not have wide expression of low-level keratin 6A. And this is mRNA contamination that is much less in the suction blister approaches when compared to biopsies. So what are we doing with this ambient RNA contamination? How can we handle it? Of course, you always need to look at biological plausibility. You know that T cells don't express keratins, and when those genes show up in your lists, you might either ignore them or you try to clean it out. When you look at this big data set, for example, here this is a lymphocyte cluster, T cell cluster is shown by expression of CD3. And you see that the CD3 expression is nicely present here. Not much is going here over there. And CD3 is obviously not contaminating all the other cells. But when you look at keratin 10, for example, which should only be present actually in keratinocytes, you see that it's nicely expressed here, but it's contaminating all the other cell populations. So what can you do? You can, of course, or when you look at those cell levels, you see that within T cells, this expression of keratin 1 and 10 is at a lower level of magnitude than in keratinocytes. So here, the expression level is around 2. Here, the expression level is around 4. And what you can do, of course, is just cut it off and get rid of this signal. And you need to be very careful, obviously, not to introduce bias here, of course, but that's one way to do it. There are now algorithms out there that can help to decontaminate your data set, that you come from this signal that contaminates all your cells to this signal where you have nice expression of keratins within your keratins, and one is DecOntX, for example, which is an unbiased approach to eliminate ambient mRNA contamination. But with all of these algorithms, of course, you always have to be very careful not to introduce bias or look at artifacts. And with any transcriptomic method, I think, especially of this complexity, you just need to confirm your major signals in an independent sample set using an independent method such as immunohistochemistry. I think there's absolutely no way around it. But when you went through all of these steps, you have very nice data sets in a two-dimensional space where you can very easily look at various markers that are being expressed in lymphocytes, for example. You can compare healthy control skin with atopic dermatitis or chronic nodular prurigo, as in this instance, and you can very nicely plot all your markers and compare those conditions and see, for example, that in AD you have maximum production of interleukin 13 and interleukin 22. One thing that you can also do with single-cell RNA sequencing, you can also perform T-cell receptor sequencing and pair your cells with the T-cell receptor information and look, for example, do you have a clonal population, do you have a monoclonal population, a polyclonal population, or an oligoclonal population? And I think clonal studies so far with rearrangement studies, or also high-throughput sequencing, has the limitation when you look at bulk tissue, you cannot really map it out. But now by combining single-cell RNA sequencing with T-cell receptor sequencing, you can nicely map the top oligoclones in a sample, like in atopic dermatitis, for example, here, when you compare to chronic nodular prurigo healthy controls. And you see that this class is really dominated by an increase in oligoclones of atopic dermatitis when you look at the top 10 oligoclones. And this is how we and others saw that you get some sort of oligoclonal expansion in the skin of atopic dermatitis patients, potentially due to environmental allergens. And this allows us now to study these cells at unprecedented depth. One great thing about all these single cell and high throughput methods is that you can analyze very small amounts of tissue. For example, in these patients, they got biopsied because they had this dupilumab-associated head neck dermatitis, and a biopsy of a few millimeters was enough to get a high level data set with lots of cells out of those few patients. And there, in this particular study, we paired gene expression with T-cell receptor expression, and saw that particularly in head neck dermatitis, as a dupilumab-associated head neck dermatitis, you get an oligoclonal expansion of top clones that are very rich in IL-22, for example, that do not express IL-26 or CSF2, and this is a population where we believe it's particularly CD8 positive oligoclonal population where we think they might react to some environmental allergens, and that might make those patients prone to this head neck dermatitis when they receive dupilumab, as opposed to the majority that does not develop this kind of side effect. But again, everything you find here, you always need to confirm it with some independent method, otherwise you might be fooled by artifacts. With this slide, I would like to conclude. I want to thank my group, all my collaborators, my mentors particularly, and the funding agencies, and thank you so much for inviting me.
Great talk. This is the first time I heard about RNA contamination in single-cell RNA-seq, and it's very educational for me. So I have two questions related to mRNA contamination in single-cell RNA-seq. So the first question is, how do we distinguish a real contamination versus some genes that are just highly expressed, for example, in keratinocyte, but have a broad expression spectrum, also expressed in T cell, but just lower expression? And I will ask the second question, if I may, relate it to the first question. So for the real RNA contamination, is there any structural properties of the RNA that can allow us to predict this is real or not? Because my guess is those RNA would preferentially bind to the cell surface. So there might be some clues we can identify based on RNA stability, 3D structure that can allow us to predict whether this is a real or not contamination.
I think the most important thing is biological plausibility. If you know genes should be expressed here or there, of course you might miss the cell that you now discover that expresses this gene, but you know that One thing is biological possibility, and the other thing, if you have mRNA contamination, it usually gives you a low-level signal in all cells, no matter what, and this makes no sense, right? So usually if you have a gene that is expressed by a certain population, you get a high peak. It's sort of a much higher expression, so keratins are very, very high in keratin, and when really all cells of your entire data set have low-level expression of this gene, that's most likely contamination, right? And when you have a lower expression of this gene in a different patient population and also the surrounding levels go down, then you're confident that you're dealing with birth mRNA contamination. But at the end of the day, it's biological plausibility.
Hi, very nice talk. The finding that you saw that the head and neck dermatitis, and you see these IL-13, 22 expressing clones even after dupilumab treatment. Any hypothesis what could be some maintenance factor or kind of survival factor for these cells? Because, when we think about Th2, we always think, IL-4/13 differentiate Th2 cells, but the drug is on board. It seems like the drug's working quite well. Any insight from your single cell data there?
Yeah, well, this is speculation, of course. I mean, when we look at the surrounding cells, like dendritic cells, they are a very good integrator of type 2 inflammation. So we have a steep downregulation of CCL17, CCL18, and all those downstream type 2 markers. So they really disappear, despite you still have some IL-13 expression. So I think that's the proof that dupilumab works very well in those patients and really blocks IL-4 receptor alpha very efficiently. One could speculate that those cells in this 10% of patients, roughly that, or less, or around that percentage that develop dupilumab-associated head neck dermatitis, they might have preceding of some type 22 cell that might react to environmental allergens. There is the hypothesis that malassezia might be here a factor, and when you get constant T-cell receptor stimulation, they might survive in that area. So I guess it's sort of a preceding of those cells that might then give you that side effect.
Thanks for a great talk. You mentioned you saw these oligoclones in the patients with head and neck dermatitis. Did you see any shared characteristics of clones between patients suggesting a shared epitope? No, okay.
No, I mean, we can, of course, it's from those data, we cannot reconstruct like any we do not know what is the antigen, and we did not see overlap in the TCR sequence, no. Or not substantially enough to make a conclusion, but those are limited numbers of patients, right?
Okay, very nice talk. And I have a question that the expression of IL-17 associated cytokines is often lost in the single cell RNA data. And you mentioned that you think the facial and neck lesions in 80 patients is about 22 inflammation. So I want to know what do you think about the IL-17 cytokines and the 22 inflammation in facial and neck AD patients? Because we know that the 22 cytokines is also included in IL-17 pathway.
Yeah, I think that's an excellent point, absolutely, because we somehow expected IL-22 producers that they might also produce IL-17, and it is true that IL-17 there are certain genes that are not very well captured by single-cell RNA sequencing. I believe IL-17 is also part of it. CD4 is also notoriously difficult to capture. You get lots of technical dropouts. So we did, from an independent sample set, we did quantitative RT-PCR to see that there was really no IL-17 expressed, and this was in line with the single-cell data. So we are pretty confident that we are not missing out on any IL-17 signal, yes.
This is such an amazing talk, and I thank you so much for giving it because it's so complex. And I was just wondering if you could tell us a little bit more about what your opinion is of the in situ sequencing platforms that we have now versus these droplet-based platforms. So something like Visium, the high-dimensional Visium. That's good.
Right. Yeah, so I mean, the single-cell RNA-seq single-cell RNA sequencing depends on cells that you isolate, and with every isolation step, the second you take cells out of their natural environment, they're going to change their transcriptomic features. And the hope for the future is, of course, that all these spatial transcriptomic methods will help us to really look at those cells in situ. I think right now, technically, we are not there yet. Just the area that's being investigated is too big. There are some other methods like MERSCOPE that looks at with in situ biased methods with probe biased assays that give better resolution. But for sure, that's the future. And I'm pretty sure that in a few years from now, everyone will do spatial and no one will do single cell anymore. Yeah. But right now, I think it's the most accurate way to look at it. Yeah.
Okay, so it's really an honor to be here again and to be back up on the stage with you. And so I'm talking to you today about IL-13 hyperpolarized inflammation in adult atopic dermatitis. And this is me and where I sort of come from in Montreal, Quebec. I work in an environment where we actually service a very large territory. So in Quebec, our university center is responsible for caring for a large population of Anglophone, Indigenous-speaking populations in our territory. And we are situated in Montreal, in fact, on lands that once belonged to our Indigenous peoples as well. And we do have a lot of considerations with these patients and these populations still in the therapeutic education that we need to give to them, given the range of dialects and languages that are spoken. And I bring this up because we know that there is so much variability in the disease. And we know that in patients like these who are still, making news headlines in Montreal and in Quebec, with outbreaks of impetigo, outbreaks of severe eczema, that we're having a hard time treating these patients, especially in these communities. And we wonder about that variability in the disease and potentially in their environments, as we've seen this morning with these remote areas and their access to water and bathing. We know now, and which has been shown so elegantly, that there is a high variability and heterogeneity in this disease, and we see that across all of these phenotypes of patients across the ethnicities. But we still have a lot of questions to ask about that variability. We want to know whether or not it's when we're catching their disease as well, because we often see that these patients may have their redness and their erythema and their inflammation masked. And in our cohort, we see that we are in an environment where we have, we do not have an exponential, what I'm trying to say is our ethnic deviation is highest in our Asian populations. So we don't see an expanded Black community coming to our clinics relative to census, but we do see this expanded Asian group coming to our clinic that is above census of Montreal. And we also see, in line with that, our Indigenous patients as well. And so we have questions about whether or not these ethnicities are related. In these patients, we can see that the impact of the disease can be very striking, and we see not only the accumulation of things like vitiligo and alopecia areata, but also these pigmentary alterations, the loss of the natural skin color, and these changes like this dirty neck that we used to say, which we're still trying to understand the mechanisms that lead to the development of these more persistent, chronic, and severe skin changes. We know in our center, we also see that many of our non-white patients actually present to the emergency room more often, and we see that when they do come into our cohort, as you see here, that these patients are also, if you look at the very severe and severe patients, there is an overrepresentation there of our Asian patients still. And as we're all sitting in this room here, I want to reflect on the fact that we have come such a long way. And so when I was, a medical student, we were still learning about wet wraps in adults, how to make sure that their topical corticosteroids would be well absorbed through the skin, and you can appreciate just how far we've come. And I think we all share this common goal that we want to seek remission and or prevention of disease. So in this talk, we're talking about this as an adult disease, wondering about this being a disease that should improve with age, and wondering why some patients do not. Why do some people improve, and why do others persist and struggle throughout their life, suffering from the itch and burden on sleep and mental health, and the immunologic factors that contribute to that increased severity and disease persistence across a lifespan. We now look at this and wonder, is IL-13 now sufficient to drive disease? We know that we can achieve remission in patients by targeting one single cytokine, one small cytokine, IL-13, and yet not its sister cytokine, IL-4, which we always thought was more important to Th2 biology. And we know that in human skin, the canonic IL-4 and IL-5 Th2 cytokines are virtually absent, or very low, even by sequencing. In contrast, we see that there's a significant increase in IL-13 in lesions, and this has been shown both by sequencing and by proteomics, and that IL-13 cell populations are found in the lesions in the blood of patients. And in particular, Dr. Gottman-Yaski and others have shown that circulating CD8 IL-13 positive cells have been found to correlate with atopic dermatitis. So these cells that are not behaving the way we think Th2 cells should be behaving, but rather these atypical type 2 T cells or TC2 cells. So while therapeutics like dupilumab taught us that atopic dermatitis is a Th2-mediated disease, what we are learning now is that we have to focus on certain cytokines within that Th2 package. And when we think about that, we start to question, well, what are the cells and how does this come to be? And we've learned now from our IL-13 targeting therapies that this is really driven by a single cytokine. So while we think about this, one of the things about studying this condition is that there are so many great questions that still remain to be answered, and really fundamental questions to the way our immune system develops. So when we think about our conventional Th2 cells, and I think anyone here who went through training in medical school will see this as being familiar with your antigen-presenting cell, being stimulated by a second signal, a co-stimulatory stimulus, then being polarized by a polarizing cytokine to develop a differentiated Th2 pathogenic effector, with this sort of suite of cytokines, which we all know are actually controlled. They're within a chromatin hub in this shared locus where we would always expect them to be expressed in conjunction with one another. But then we start to see things breaking apart a little bit. We've talked today about this oligoclonal cell expansion. We know that others have found it's really polyclonal, and we still are looking for those specific antigens that may be driving this disease, but we haven't really found them. And we know that it's a cytokine that sort of expands above and beyond and does essentially its own thing in these cells. And we wonder about what antigenic trigger that could be driven by. And especially given the fact that, as we've learned about this IL-4 presented by the antigen-presenting cell, that we don't see IL-4 presented by antigen-presenting cells in the lymph nodes, that the dendritic cells do not produce this cell that is this cytokine that's so important to generating these Th2 cells. So we like to ask questions about these cells and these cells in particular that produce IL-13. And we like to even postulate that the way we've understood our immune system through these traditional lymphoid organs, secondary lymphoid organs, may need to be revisited as we're learning more and more about the immunology of the skin through all of these advanced technologies that we have now, and as we are essentially manipulating the immune system with our therapeutics. And the proliferation of therapeutics to date, changing the immune system of our patients, and for many patients, potentially across their lifespan. We also know that these T cells in atopic dermatitis start to acquire these innate-like properties when they're found in the skin, where they can express these receptors that we typically would expect to see on our antigen-presenting cells. And so we wonder how these cells come to be and what their role is in producing the cytokine IL-13. And we all recognize, and have talked about this already today, the role of Staphylococcus aureus in the surface of the skin. Knowing that it can readily penetrate skin through paracellular as well as intracellular routes. And Dr. Gallo is in the room and has taught us so much about all of the interactions of Staph aureus and our innate immune system and the role it can play in atopic dermatitis. We know that that's well situated to be triggering our alarmins and generating these more innate lymphoid cells in the skin. And we know that these alarmins are really essential for this non-humoral type 2 inflammation. If we get rid of alarmins, we can still develop humoral Th2 inflammation in mice, so IgE-mediated responses. But we have a gap in developing these cell-mediated effectors. We know that there has been an enormous body of literature and essentially a scientific wave of progress through our knowledge of these innate lymphoid cells that reside around hair follicles and in tissues with high microbial loads. And in particular, we see that they themselves express these receptors that allow them to respond within seconds to minutes of danger or stranger signaling in the skin, where mast cells can become activated and they can respond to those mediators through receptors like CRTH2 responding to a prostaglandin. And that these receptors for the alarmins and their co-stimulatory receptors can essentially license them to develop these IL-13 responses very quickly. But we haven't seen in the single-cell sequencing, we have not seen such a prominent role for human innate lymphoid cells in atopic dermatitis skin. So ILC2 exists, but are they really elevated to the same significant elevation that we see Th2 cells relative to healthy subjects? Remains, I think, an ongoing and active area of debate. And so we ask ourselves whether or not the cells in atopic dermatitis patients are a little bit of a different type of cell, so maybe more of an innate-like T cell, which express many of these same receptors, it appears, and may be able to effectuate the same responses. And so the question is really, what can drive that IL-4 independent transcription of Th2 cytokines, the way that we see them developing in innate lymphoid cells. And we know that there is a whole host of different cell types that can be contributing to this, including innate lymphoid cells, CD8, intraepithelial lymphocytes, NK, NKT cells, and myeloid cells, a population family that we're still just trying to get to know. So when we think about these tissue-specific IL-13 cells in the skin, we may want to look at the skin and say, well, maybe we need to think about another source of these cells and look back at where these cells are coming from in early development, certainly given the fact that this is a disease that develops so early in infancy. And we know that the skin is populated in fetal development and in early neonatal life by cells that are generated out of the liver, out of the bone marrow, and that the signals that we've seen with these increasing chemokines, these populations of T cells that are homing to the skin so early in life in our infants, are really developing at a stage at which our secondary lymphoid organs themselves are not well developed. So how does this lead to our populations in skin? Without these cytokines, we think about, well, do we really only want to focus on this Th2 cell? We need to look at these other cell types as well. We know that there may be CD8 and others producing this super polarized IL-13 response. But we want to know, like, what are these innate lymphoid cells? Are the CD8 IL-13 producers pathogenic in AD? And we know that there is such an enormous and rich knowledge that's come out of our single-cell data sets to date, but we've also heard about the limitations. We know that there are few samples and few patients, that most of these patients are white, and that there's still a paucity of studies of skin in situ. And so the in situ cellular biology of this disease remains an open forum for investigation. And we know then as well that skin structure leads to important limitations in the detection of proteins in particular in the skin. And so that to date there's been limited cell-specific cytokine localization at the protein level. And we know that although all of these new sequencing data sets have come out, we know there's also variability between bulk sequencing to single-cell sequencing results, and even from single-cell sequencing to single-cell sequencing results, which just may be due to the variability of the disease itself, the stage at which it was biopsies, the location, the patient's ethnicity, et cetera, but may also be due to the technical limitations themselves. And so we do need to pursue innovative approaches. So in our lab, we're interested, of course, in characterizing these IL-13 superpolarized cells. And we focus on the tissue-resident memory cells, of course, believing that they may impair remission. And we really seek to characterize these IL-13 high CRTH2 marked pathogenic effectors in AD skin. And because we work with this range of ethnicities and we have this large biobank of skin that's been collected, we're looking at methodologies that are a little bit more agile for hypothesis testing in situ. And so we focused on the use of confocal microscopy, and in particular looking for these populations, the proteins, using both protein and RNA-based approaches, which we'll look here. So in general, we know that IL-13 protein detection in the lesional skin of AD patients does vary quite significantly, but that it really does concentrate in that epidermal layer. And the work that we do here quantifies protein expression across the entire 5 millimeter punch biopsy tile scan. And we also can break this down into the epidermal and dermal compartments and quantify each separately. And so this is just the overall study design that we've used, with obviously a limited number of patients here, but also indicating the ethnicities that our patients have come from. And if you look down towards the clinical scores, you can see that we really are working with a severe cohort here from what I'm showing you today. And I've shown the picture on the left before, where we know that we have this incredible IL-13 detection in skin. But I'm now showing you also what we were finding in the skin for IL-4, where you can see that there is detectable protein for IL-4 that we can find in the skin of patients. And this is what we've looked at quantifying here. So below you can see that we're demonstrating IL-13 protein as a fold and IL-4 protein as a fold increase over healthy skin, with IL-13 in red and IL-4 in green. And what you're seeing here is the striking increase in IL-13 protein, not only in lesional skin in red, but also in non-lesional skin right next to it. And you can detect that this is quite different from what we see with IL-4, both at a concentration level as well as using a weighted quantification across that entire tile scan, accounting for the area that's been imaged, given the difference between lesional and non-lesional thickness of the epidermis. And so this increase in IL-13 signal in the non-lesional skin of patients is something that at the protein level has not been reported, or that there may be limited studies to date looking at that. And so we're quite interested in the fact that even the unaffected or non-lesional skin of patients can have this really important signal. And we see that it's really concentrated in that epidermis, both in the lesional and the non-lesional skin, but also is found in the dermis at lower levels. Oops, I'm going to go back here. Well, we've just skipped ahead on the IL-4, and I don't know how to work this backwards. Thank you. Thank you. And so here we have IL-4, and you can see that, in line with what we've seen with proteomics, right? So, in proteomics, we see that IL-4, you can see this increase, but it's not significant. And you can see that here with the concentration of IL-4 on the left-hand side in the total skin, but you can detect, a trend in the lesional skin when you break it down into the epidermis and dermis. So we do think there is a signal there. We just don't find that it's significant compared to IL-13. And really, we're looking at this fivefold higher concentration of IL-13 in both lesional and non-lesional skin. Significantly increased in both of these compartments, about fourfold higher than IL-4. And in particular, we think that this increase in protein in the non-lesional skin is consistent with this IL-13-driven non-lesional itch that patients complain of and that we've heard about today. So what about the cells? So here we've gone on to quantify cells, again, comparing IL-13 producing versus IL-4 producing T cells staining for these proteins. And on the left, you're seeing the full thickness skin. So I've just indicated above what the full thickness looks like. And then as we break it down into the T helper cells and the T cytotoxic cells across this full thickness of skin, we can see that, in fact, the IL-13 positive T cells are increased as we would have expected them to be, and that here we do not see this increase in significant IL-4 producing T helper or T cytotoxic cells in the skin. When we start to look at that broken down into compartments of the skin, we see that we have a pretty bland-looking epidermis at the protein level in terms of these T helpers, but that in the dermis we see what we would expect with an increase in IL-13 producing cells in the lesional skin that's significantly elevated, significantly more than what we see in the non-lesional skin as well. Again, with our favored cell, these cytotoxic T cells, now if we look at the epidermis, really this is the target, right? The epidermis is where all the biology is happening in our atopic patients. We see that there's an increase in these IL-13 positive cytotoxic cells, and we see that they're increased as well in the dermis, again, showing the IL-4 population flat. So in summary, for this section, we see that these IL-13 but not IL-4 positive cells are increased, and they are specifically increased in that lesional epidermis. And so this argues for the unique role for these non-canonic T cells in this hyperpolarized IL-13 state that our patient's skin exists in. So we moved on to look at the expression of these cytokines and to compare them, knowing that IL-13 can be readily bound to protein, and a lot of that IL-13 that we're seeing in the skin is, bound to keratinocytes and other cells. And so what I'm showing you here is an approach that we've used to looking at in situ detection of target. And we're using this technique, RNA-scope, in situ, and we've just shown you here the panel that we used to test some of our hypotheses about these atypical cells, looking at, of course, our cytokines of interest, but also some general lineage markers, as well as OX40 as a favored activation marker and co-stimulatory receptor that's now druggable, as well as CRTH2 and our tissue-resident memory T cells, and these markers of pathogenic effector cells or innate lymphoid cells for some, the TSLPR, as well as the GATA3 transcription factor. And so we started off by just describing, like identifying, can we find these cells and detecting them in the skin? And then we moved on to working with a pipeline for informatics that we imported from the neurology space to process this data and to quantifiable data that we could then analyze in what isn't big data, but that is still data that's beyond the scope of my own brain to compute. And so here, though, what I'm showing you are just sort of simple analysis before getting to that unbiased approach. So we're just looking at the fact that, as you see, when we break it down into the compartments, we see that the atopic dermatitis epithelium, marked here with just an E, is really where all of this business is happening, where all of these specific targets are highly expressed. And we see that if we look at IL-13, I'm not sure, oh, for some reason it's not projecting properly. Oh, that's too bad. So this is, you'll just have to believe me, and I can show it to you afterwards on my own screen. But we see is that although you can detect IL-4 in the epidermis and in the dermis, you really only see a very low-grade expression. So the intensity of that signal is very low, whereas you can see this very high range of expression for IL-13. And so here what we're demonstrating is the value that we see in this methodology, which can allow us to not only quantify these cells, but then to plot them spatially using these X and Y axes into the skin. And I'm showing you the identification and annotation of these cell types that we're interested in. So at the bottom are tissue-resident memory T, in green are non-T tissue-resident memory cells. We've also identified this population in light purple lavender of pathogenic effectors using the marker CRTH2. And we're also looking for cytotoxic tissue-resident memory, and we even have a population here of double-positive tissue-resident memory T cells as well. And that what we're doing afterwards is just you see overlying the expression of these cytokines, so IL-13 versus IL-4, with those populations of interest. And we can analyze this data using this U mapping approach and actually then treating it almost like flow cytometry data. And what we've been able to do with that is to look for these particular cell types that we're interested in. And this is now data that's coming out from the cells that we're quantifying, so just gating on these CD45 TCR positive cells in the epidermis, for example, and showing that there is an increase in these CD8 T cells here at the RNA level, but we're actually seeing more of a significant increase in the double positive CD4 and CD8 T cells. But when we focus in on our cells of interest and look to see, what are these cells expressing, how are they characterized, what we see is that our cytotoxic T cells indeed have this IL-13 high, IL-4 low signal, that they are tissue-resident memory, and that they have low expression of CRTH2, but quite significant expression of OX40, as well as a low expression of this TSLPR as well, indicating that they can be readily licensed in the tissue to respond potentially independently of any secondary lymphoid organ, responding rapidly to triggers that we see in the tissues. And in the epidermis as well, what we're seeing is that this is really where we see these CRTH2 and OX40 positive cells. They're much more significant in the epidermis. Even though this is a small group of patients that needs to be replicated, we're seeing that this is where, again, these pathogenic markers are acting. So in summary, what we see is this superpolarized IL-13 producing cells are accumulating within the epidermis. And this is, of course, the most disturbed skin compartment, so we're really interested in digging into that compartment and understanding more specifically what's happening within that tissue microenvironment. We know that these activated epidermal cells with this unique IL-13 high, IL-4 low polarization state are CD8 as well as non-T effectors. And we see that this is really driven by these non-canonical cells in the skin niche, and yet the mechanisms for driving that IL-13 production from these atypical cells and CD8 cytotoxic cells really still remains unclear. We also don't know whether or not this will be true across all ethnicities. We don't know how these cells develop, and we need to focus on identifying whether or not these novel pathways contribute. And so, of course, our ultimate goal is to see whether or not these skin-specific cells can be eradicated. And I just want to thank my lab and my funders and your time and attention for listening to me this morning, or this afternoon, or this morning, still this morning. Any questions?
I think we have time for one question.
Okay. I'll be around afterwards if there are any. Okay, so at this time, it's my pleasure to introduce Dr. Parada. I'm just going to grab my notes. So Dr. Paraga, Shirley Paraga, is coming to us as a research fellow from Wake Forest University School of Medicine, and also trained at the Brody School of Medicine with Dr. Koster. And she is now coming to us to talk to us about her interest in artificial intelligence, along with her interest in clinical trials, and in particular, looking at facial characteristics, which is a really fascinating area to be looking at. So thank you very much, and we look forward to your talk.
Alrighty. Let me make sure I have, yep. Okay, so thank you, Dr. Jack, and thank you to the International Eczema Council for allowing me to present our my research team's collaborative efforts today. There we go. Institutional support for this study was provided by the Perfect Skincare Pro Corporation. I myself have no conflicts of interest to disclose today. Alrighty, so as we know, atopic dermatitis is an inflammatory skin condition with a chronic relapsing nature that commonly affects the face. Face rashes may present as extremely pruritic, erythematous excoriations. However, additional clinical features of chronic atopic dermatitis may lead to other skin changes, including erythema, flaking, scaling, lichenification, or hyperpigmentation. Additionally, skin involvement around the eyes is very common in our eczema patients. Eyelids may become thickened, dark, and wrinkled. We've also found that lower eyelid creasing or Denny-Morgan lines are common development in atopic dermatitis, and it's often due to the repetitive rubbing and scratching due to the intense pruritus and chronic edema in the infraorbital areas. This is a feature that's present in up to 25% of our eczema patients. So because facial involvement in eczema presents as a particularly bothersome issue for our patients due to the high visibility of the disease symptoms, we wanted to see if we could incorporate the use of AI. We've also noted that it can be difficult to treat the face as it may respond to therapies differently than other body parts. And then this is where facial AI systems come in. So by helping us monitor the progression of eczema through analysis of specific facial characteristics, including wrinkles, redness, texture, spots, dark circles, overall skin health, and others, we believe that we may be able to help patients identify and establish baseline metrics, and then from there on gauge whether or not their treatment response is working appropriately. So for our study, we evaluated the validity of a particular facial AI system, and we used it to detect changes in facial characteristics for our eczema patients. So the way we conducted our study is we recruited 10 patients over the age of 11 that were starting a personalized eczema treatment, and these were patients from the Atrium Health Wake Forest dermatology clinics. We took multiple images using 2 facial AIs, one being the Perfect Skincare Pro, which is an iPad-based AI, which I'll provide more information in the next slide. And then we also utilized a computer-based model, which is the Vizia, and this is traditionally considered the gold standard in clinical facial assessments. Additionally, images for both of the systems were taken in a clinical photography room under standardized lighting, as you can see in the image to the far right. The way Perfect works is it provides us with a raw score, and the lower the raw score, this indicates a less desirable skin characteristic. So we chose to analyze the progression of skin characteristics that are most commonly involved in manifestations of facial eczema. So for primary outcomes, we analyzed the changes in facial redness, and secondary outcomes, we analyzed periorbital and under eye involvement. So we focused on eye wrinkles, eye bags, and dark circles. And then these changes for the characteristics we chose were compared at baseline and then 8 weeks after initiating treatment. And then in addition, we also performed separate physician assessments for the same characteristics during the clinic visits, and then compared these scores to the scores produced by the Perfect AI. Alrighty, so to go a little bit more in depth into the AI interface, it's an iPad-based system with iOS compatibility, and it also does rely on Wi-Fi for algorithm processing. So what makes Perfect AI unique is that it incorporates the deep neural network processing algorithm, which is designed to process external inputs, analyze patterns, and predict outputs for 14 facial characteristics, which include brown and red UV spots, redness, wrinkles, pores, moisture, oiliness, texture, dark circles, eye bags, and then droopy upper and lower eyelids, among others. And the DNN algorithm has been trained in the input of 70,000 images from faces of represented by diverse ethnic backgrounds. So as of last year, it was validated across Fitzpatrick skin types 1 through 6. And then, as you can see on the image on the right, this is an example of the assessment produced by the AI. So the way it works is either the patient or the clinician will hold up the tablet, and once it's aligned properly, this provides an instant analysis. And then it provides us with a raw score for each of the 14 characteristics, which is shown on the bottom of the assessment there. Alrighty, so these were the results for one of our patients who presented with particularly pronounced infraorbital involvement, as well as several erythematous and scaly patches throughout his face. It's a little hard to see, but as we were able to note, after 8 weeks of topical treatment, his raw score significantly improved, indicating that his treatment response was appropriate. So the improvement was primarily noted for redness, which increased from the 79th to 89th percentile. Eye bags increased from the 73rd to the 78th percentile. And eye wrinkles and dark circles, which were our 2 secondary outcome measures, stayed the same at 84 and 68 percentile, respectively. And then we observed similar trends in the majority of the patients that were enrolled in our study. So the changes in mean raw score for facial redness indicated a statistically significant improvement from baseline to final visits at 8 weeks after initiating treatment. And this is for facial redness. While raw scores for eye wrinkles, eye bags, and dark circles did improve on average after 8 weeks of treatment, they did not produce statistically significant results. And then finally, through comparison of the perfect AI and physician assessments, we were able to conclude that there was a mean concordance rate of 70% across all 14 of the facial characteristics analyzed. However, for the specific skin characteristics we focused on for this study, the agreement rates were highest for redness, eye wrinkles, and eye bags, all three of which produced a statistically significant value. And then the lowest of the agreement was for dark circles, being 59%. Alrighty, so overall this study indicated to us that facial AI technology can capture changes in facial redness and under-eye skin involvement, and it proved to be a fast, practical, and very useful tool, something that patients and clinicians can use to monitor long-term and hopefully within and not necessarily a clinical setting, but rather a home setting as well. We found that using this AI technology generated reliable scores and may be a valuable research and clinical tool for dermatologists to help assess baseline metrics and then track skin concerns for eczema patients, as well as determining if treatment response is appropriate or if alternatives need to be considered. And these tools were convenient, user-friendly, and cost-effective relatively compared to the traditional used models. These are my references. And then we would like to acknowledge both the CTSI department at Wake Forest for sorting us with the data processing, and then Dr. Feldman's Center for Dermatology Research Center at the School of Wake Forest and all of our team. Thank you. I'd be happy to take any questions.
Hi, thank you. Very interesting. I'm curious, I guess, two things, whether you have tested this not just using the iPad, but across multiple cameras, devices, because you mentioned the ability to do at-home collection. I'm also curious whether you're aware if this technology has been used at all for vitiligo studies.
Yep, so that's a good question. So and this is why we did incorporate the Visia model, just because, like I mentioned, this is the gold standard. I know there are a couple of new AI systems coming onto market. For now, to my knowledge, Perfect itself, the portable iPad device, has only been validated for rosacea and melasma. We are actually hoping to validate it for acne by the end of this year, and I'll be presenting on that one shortly after, yeah.
It's great to see the convergence of all of these approaches, right? And to be able to look at imaging in these innovative ways. I was wondering if you could speak to the detection of erythema a little bit, and the population that you had, and how diverse the patients were that you had, like just their skin phototype of yours. And just to ask in particular, how do you validate erythema detection? So to expand on that.
Yeah, so that's another interesting question. We actually did find some discrepancies depending on our patients' Fitzpatrick skin types. That is something that we are still trying to establish, whether specific Fitzpatrick skin types analyze the erythema as well as others. So the study that validated the Perfect system did say that it had a highest agreement rate for Fitzpatrick skin types 1 through 3. So I think that's where additional research needs to be conducted. But yeah, that's a very interesting question. All righty, thank you.
Okay, and so it's my pleasure next to be introducing Austin Moon. Dr. Austin Moon is coming to us from the University of California at San Diego. And he is a research fellow in the clinical trials unit in the Department of Dermatology. And he works with both Dr. Hatta as well as Dr. Gallo. And his current research is focused on bacteriotherapy in atopic dermatitis. Thank you so much for coming to speak with us today.
All right, good morning, everyone, and thank you, Dr. Jack, for the wonderful introduction. And I would like to thank the IEC for this kind invitation. And it's my greatest honor to present to you my research on the effects of commonly used topical moisturizers in the survival of Staphylococcus hominis A9, a key contributor to the atopic dermatitis microbiome. And I would like to clarify that there is no financial conflict associated with this study. And I would like to start by highlighting the importance of topical moisturizers in the therapy for atopic dermatitis patients. So the AAD released a new guideline last year, and actually includes many of the distinguished authors present here today. And it's widely recommended for atopic dermatitis patients for safeguarding the skin barrier, as well as reducing the signs, inflammation, and the symptoms in patients with atopic dermatitis. And the effects of these moisturizers are commonly assessed by measuring the transepidermal water loss, the EASI scores, itch levels, and then the hydration levels, but typically not by studying the skin microbiome. And there have been many studies in the past to study the link between early topical moisturizers and the prevention of eczema in infant populations. However, there has been a variability and no significant correlation has been found comparing to the control group, but they also noted that there may be a benefit in the high-risk populations by potentially delaying the onset of atopic dermatitis. And another study, this was a meta-analysis done, looked at different, sought to explore something similar if preemptive use of topical moisturizers could prevent the incidence of atopic dermatitis in infant populations. And as you can see on the right side, there's no consensus and a variability in the outcome, meaning that each moisturizers have a different effect and not lotions are created equal. And switching gears, I would love to talk about the skin microbiome, which has been a hot topic in the field of dermatology for the past few decades. And as you may know, Staphylococcus aureus is increased in our patient with atopic dermatitis, highlighted by Dr. Layden et al. In the 1974 paper. And the pH of these AD skin is found to be more alkaline than acidic, which the S. aureus loves. And coagulative negative staph species, CONS for short, that produce antimicrobials are found to be decreased in patients with atopic dermatitis. And similarly, CONS species that produce auto-inducing peptides that inhibits the SA toxin are also found to be decreased in atopic dermatitis patients. And one strain in particular among the commensal bacteria found in the Gallo lab is Staphylococcus hominis A9. And this species is special in the sense that it produces two lentibiotics that serves to inhibit the S. aureus species. It also produces auto-inducing peptides that inhibits the S. aureus toxin. But also, it doesn't inhibit other CONS species in the microbiome, playing an integral and a balanced role in the skin environment. And combining what I talked about regarding the moisturizer and the skin microbiome, we sought to examine the benefits of moisturizers on the skin microbiome as an additional method to further assess the moisturizer properties. And we hypothesized that there we'll see a variable degree of Staphylococcus hominis A9 survivability across different lotion products. And 10 moisturizers were selected. I'm sure each and one of you have your own favorite, but these were based on how commonly they were prescribed or the accessibility and their involvement in the prior studies regarding other eczema studies. So we had 3 parts to our study. Starting with part A, we had a concentrate of SHA9 mixed with a topical moisturizer, gently mixed in a sterile tube, incubated at 30 degrees Celsius, and then samples were taken at 0, 5, 12, and 24-hour time points, and then were plated on agar plates overnight. And we found that, as you can see on the left side, 6 out of the 10 moisturizers had what we call an intermediate effect, meaning they were somewhere between bactericidal activity, but no overgrowth of bacteria. And 2 out of the 10 products, which were Vanicream and Curel, had a bactericidal activity, meaning that by hour 1 and 5, we could no longer see SHA9 survivability. And 2 out of 10 had a bacteriostatic effect, which were Vaseline and CeraVe Daily Moisturizing Lotion, which had a somewhat slow downward trend, but no overt bactericidal or overgrowth of bacteria. And part B of our experiment, we sought to explore if the pH level played a critical role in the survivability of the SHA9 strain. So we took our moisturizers, placed them each in individual containers, measured the pH, and using the data from the method A section, we sought to plot the correlation between them. And as you can see in table 1, we found that Vanicream was the most acidic at 3.4, double base gel the most basic at 6.8. Nonetheless, all moisturizers were found to be acidic. And when these were plotted against the data we obtained from method A, we found that by 5 and 12-hour time point, there was a statistical significant correlation between the pH level and the survivability of SHA9. We weren't able to appreciate statistical significance at the 1-hour time point. However, we speculate that the pH level might take some time before it starts taking effect. And finally, part C of our experiment, we sought to explore if moisturizers had an effect on the commensal bacteria on the human skin. So for each patient, we identify a specific region in the body where we had no lotion and swabbed at 0 and 5-hour time points, plated on agar plates. And another body site where we applied 1 fingertip unit of either Curel or CeraVe moisturizing lotion, swabbed at 0 and 5-hour time points, and then plated again. And the reason we chose Curel and CeraVe moisturizing lotion was that Curel in the in vitro experiment had a very strong bactericidal activity, killing SHA9 at 1-hour time point, and the CeraVe moisturizing lotion had a bacteriostatic effect in the in vitro experiment. And what we have found that is compared to the no lotion, CeraVe had an increase in the bacterial commensal population, whereas Curel had a downward decrease in the bacterial population across 5 human subjects. And our next logical step was to further explore if the ingredients played a critical role. And this table lists the number and type of antimicrobials we found in each moisturizing product, starting with the most potent at the top, which were Curel and Vanicream, and the least potent, which were CeraVe Daily Moisturizing Lotion and Vaseline in the bottom. And one thing we want to note is that between the Curel and Vanicream, which had similar ingredients to the CeraVe Daily Moisturizing Lotion, although they did share the same ingredients, they had very variable effects on the SHA9 survivability. And further assessment was met with a challenge, as the formulation and concentration of these ingredients are known, as it is specific to the company. However, we wish to explore this further into details in the future studies. As a summary, all moisturizers are not created equal. We saw a variability in the SHA9 survivability, and pH level could have a variable effect on the bacterial survival, and as well as the moisturizers can have a variable effect on the commensal bacteria population. And 2 lotions that we thought were interesting was, one is Vanicream, which is widely considered to be less irritating, was actually found to be more bactericidal than we thought, and amlactin, which is considered to be more irritating, actually didn't have that strong of a bactericidal effect as we expected. And take-home message for today, on top of considering the hydration level and protecting the skin barrier and protection from the environment, we want to plant a seed in your head, maybe considering the skin microbiome in the future when you recommend moisturizing products to your patients with atopic dermatitis. And we hope that this study will be a stepping stone towards future infant prevention studies, as they haven't explored the skin microbiome as robust as we have hoped in the past. For future steps, we wish to study more moisturizers across more human subjects, look into the ingredients more into details, and link the relationship between the ingredients and the survivability of SH9 as well as the commensal population, but also expand our research to also involve Staphylococcus aureus. And I'd like to give my special thanks to Dr. Tisa Hara, Dr. Teruraki Nagatsuji, and Dr. Rich Gallo for supporting this project and being our great mentors, but also to our members, Alex, Victoria, Selena, Katie, and Tegan. Thank you. And I'll take any questions.
Thanks for a great talk. I was just wondering, because I can't remember that slide you showed where you nicely summarized all the trials on prevention of AD by moisturization. Did you find any correlation between the outcomes of those studies and the bactericidal efficacy or effects of your moisturizers?
Unfortunately, no. Some of the products that were listed here were not included in our study, or we expand the moisturizers by having additional moisturizers, but we didn't really necessarily find a correlation between that. And this is a very early stage in the study where we haven't really went to a deep analysis of the correlation between the commensal and the effects of moisturizer, so that's something we will hope to do in the future.
As you told, these creams are bactericidal and bacteriostatic. Will it not disrupt our natural microbiome of the skin?
I'm sorry, can you say that one more time?
These creams, moisturizer creams, are bactericidal and bacteriostatic. So we continuously using it will not destroy our natural human microbiome, skin microbiome.
So that's also that we will love to explore a little bit more in the past, as these moisturizers were only applied once, a one fingertip unit, and only sampled at 5-hour time point. But as you may know, when we recommend these moisturizing product to our patient with eczema, they either put it twice or once a daily, constantly across many weeks. So that will be a very interesting field of research in the future to see if we'll have a long-term effect on your skin microbiome and maybe find a shift in the balance in the microbiome in the future. But that's something that's also in the books that we hope to study in the future.
Thank you so much for a beautiful study and for tackling a really challenging and complicated question for our patients. And I was wondering, for these patients, maybe if you would consider telling us what you would recommend to a patient at this point, and also for the industry itself, and particularly the corticosteroid creams and all the new topical agents that we have delivered via topical means, what your opinion would be on the base for those creams moving forward.
So this is where the study of the Staphylococcus or aureus study will be critical, because the ideal moisturizer will be for the selective growing of the SH9 and the the beneficial, quote unquote, commensals and the preferential killing of Staphylococcus aureus. So at this point, I think it's too early to see exactly pinpoint which moisturizer will be beneficial unless we have those data. But like you mentioned, the topical steroids also play a critical role too. So I think that's something of interest for us too, to see if the topical steroids itself could influence these commensal bacteria and how it plays a role with the SH9 and Staphylococcus aureus in the future. Thank you.
Yeah, let's get all the speakers. I think we need one more chair.
Can we get all the speakers up here, if that's okay?
I don't mind standing.
Where is Sarah? Is Sarah one of the speakers here? I don't know. There she is. I think one more chair. One more? Sorry. I misspoke.
No, I can stand. I don't mind standing. Just like do some jumping jacks and see. It's so cool.
No, that's okay. You know what? I will be here to sit. No, I think we are good. I'll take it from here. So we'll welcome any question.
Do you have any
Don't be
questions?
shy.
Any questions for our panelists today? Raise your hands. I'll come around with the mic.
See, everything was super clear. No one? Yeah. I see one question here.
I have a question.
Ah.
I know that there's just a lot of exciting new therapies out for atopic dermatitis. What do you really see as the future kind of unmet need that we should be tackling as new targets now?
Yeah, so the way I see it, I think is twofold. First of all, I think in all patients, we need treatments that will be disease modifiers. And, you know, I know it's a big a description, disease modifier, because dupilumab, for example, was also a disease modifier, right? And it is to some extent. But real disease modification, I think, is the ability to either take a treatment every 12 weeks or maybe even more, or even to take it for a while, let's say 1 or 2 years, and then be able to stop it and not need a treatment again, or maybe in a few years, you know, not. Because I think the number one thing that patients are asking us is, doctor, do I need to take this treatment for life, right? They do not want to take that treatment for life. That chronicity of treatment is a hard concept for them to accept. And I think we also need treatments that early on will be able to prevent the atopic march in children that are in high risk for it, or to prevent other atopic conditions when a child already has one of them, right? Because the rule of thumb is if you have one, you'll likely get others. That's my thought, but I welcome others here. Anyone? Okay. And I think there was a question here too. Over here.
Well, I'd like to add that actually Dr. Gallo and Dr. Tisa are doing starting an exciting study soon where we have implant these beneficial SH9 therapies to the patient with eczema. So hopefully we have high expectations, and then hopefully this will lead to a new therapy in the future for treatment for atopic dermatitis.
I think to your point, to the point of I agree with Emma, but to the point of specific issues, there are a lot of unmet needs, and one of them is, but still I think we need topicals that are better. I mean, yes, biologics and targeted treatments. To that point, addressing the microbiome would be very interesting, and also orally, I think that even addressing orally the microbiome would be interesting to look at long-term on issues of prevention or disease modification. So this is an exciting, and I like the idea of pH. I started my career in pH of the skin maybe 30 years ago, and it's more than 30 years ago. I think my first study was skin pH, and it's unbelievable that still in the US, companies are not asked to regulate their pH products. I think the buffer capacity of the skin is so strong, and the acidity is so helpful for, I wasn't aware of it. Low pH actually reduces itch because activating the serine proteases activate part 2 of the non-histaminergic pathway in alkaline environments. So there's a lot of things to learn. There's a lot of things also to learn about the specific phenotypes in populations. I think to Emma's acknowledgement, she studied not just in skin of color, but in Asian skin. I think that we're not yet clear about what's the best treatment for each population. So we can talk a lot about these issues, but I think...
Yeah, I just to say, I completely agree. I think the next frontier in atopic dermatitis is to treat better or better designed treatments for specific populations, right? Because right now the treatments we have, I'll give the dupilumab example, EZ90 is about a third, right? And that's about right also with other biologics as well. And I think the reason being the heterogeneity of the disease that is so large, and we need to treat better skin of color, to also recognize it better, right? The diagnosis of where lesional skin ends, non-lesional begins, and so on. And there are some attempts to do that. For example, I'm sure you guys saw the study from Eli Lilly at the AAD. And I think more studies need to be done to really target specific populations. Yeah.
Yeah, and I just wanted to expand on that, actually. I think, one of the thoughts that I have is that, I think in the future, too, we'll be thinking completely differently about the way we train our immune systems as well, and I think that in a lot of this, it's about developing tolerance, and I'm kind of curious to speak about this with Dr. Gallo in the room, thinking about the role of Staph aureus and whether or not we should be inducing tolerance to this microbe, or how to approach that vaccination strategy, maybe through alternative routes of the gut mucosa as well. And so lots of questions about how the immune system can develop, because I think the earlier we can interfere, the better.
Yes, I agree 100%. Early intervention will be key here, because once those, especially T cells, are all in those end organs, they're very difficult to get rid of. But I think it's not impossible. I mean, we can extrapolate from cutaneous lymphomas, for example. With eczema, it's often difficult, the egg-hen issue, but in cutaneous lymphomas, we know a malignant T cell sits in the skin, and we found that in normal appearing lymphoma, you get a lot of malignant cells, but they seem to be silenced. And if we cannot get rid of these T cells, I think silencing them in the skin with disease modification, that's a holy grail, of course. And I think from lymphomas, which can sometimes cause eczematous lesions, I think we know that this might theoretically be possible.
Right, and also I think in the future we need more studies like the one Patrick did, for example, showing the facial eczema, which patients are more likely to get that manifestation, so that we really think about which drug for which patient based on their unique characteristics. Yeah, we didn't forget about you.
Jim Park from Mass General Hospital. and this question is mainly related to Dr. Yoshipovich's presentation, but also related to other speakers who presented multi-omics analysis of AD skin. So neuroimmune interactions emerged as a major topic, particularly in the space of atopic dermatitis, and emphasis initially was on itch, but now AD and inflammation per se appears to be driven by the peripheral nerve activities. So most of the literature, information from the literatures and studies focused on nerve subtypes deconvoluted with multiomics in DRG, not actually the skin nerves. So there are no shortage of subtype-specific markers that clinicians and basic scientists use based on DRG profiling, but I was wondering whether the same marker sets can resolve the skin nerve fibers that actually derived from DRGs, and then what are the challenges in gaining most of this information, because most information from the literature uses pan-neuronal markers like a PGP 9.5, which really doesn't resolve any specific details. So I would appreciate your thoughts about it.
You're perfectly correct. There are challenges, so there's a lot more to do. But, and I agree with you, I mean, the DRG, that's the problem, because the RNA material is in the skin not conveying what the DRG, but on the other hand, their neuropeptides and keratinocytes are also, to me, a narrow structure. So we could collect some information about that, and clearly there are specific CNR fibers and their subtypes that we could have a look into. So there's a lot more to explore in that field, but I agree that the fact that we don't see doesn't mean that they're not active, just because in the DRG a lot of activity happens. So very difficult to take DRGs from humans, not impossible. And with due respect to my studies, there's a lot of gaps there. I have to admit that it's more difficult to do, but we've done work and others did. Dogs are a very good model for humans. Sometimes the owners look like the dogs, or the dogs look like the owners in terms of their manifestation, because atopic dogs are very similar to humans, and they could be, it's not easy to do this research, but it would give us a lot more information, and they're actually very advanced in their understanding of neuroimmune. So we need to. Monkeys, primates is also very challenging, but they are very similar to humans. So we have to do more research in that area too.
Definitely. I actually have a story about one of my patients that has severe atopic dermatitis. He couldn't afford, he didn't have insurance, so he couldn't afford a JAK inhibitors, but he was buying for the dog a JAK inhibitor. Was $100 a month, and he was dividing the dose with the dog. This is, it's a real story.
Yeah, I have an atopic dog, and she got a JAK inhibitor, and she also got an IL-31 blocker. They were well ahead from the humans. Yeah.
Yeah. Right.
Of course, you can maybe tolerate a little bit more side effects in dogs than in humans, but that's a different story. I think this conversation has been fantastic. I think perhaps it's time to point out some of the unmet needs in terms of research towards atopic dermatitis. And what we're learning is that the deeper we look into our own niches of scientific expertise, the more we're observing that each of these niches likely plays a role in the development of these different populations, whether you're looking at genetics, transcriptomics, microbiome, neurobiology. What I haven't seen yet is more computational methods to try to integrate these different data sets into a comprehensive model that can predict outcomes. I'm aware of some things like this in psoriasis that are actually starting to get kind of interesting, and I wonder if any of you have seen such models.
We are in the process. We have some talented bioinformaticians at Sinai, and we now are in the process of putting together exactly what you say. So more will come.
Emma, I'd like to also point out that I think that we need to address also dendritic cells more seriously. I think the OX40 is opening that field, but there's a lot more. It's kind of upstream of representing a interaction between the environment and the allergens and the irritants and everything. So I'm looking forward to see what you guys will do, but we're looking into it, but love to hear others.
Yeah, no, definitely. I think we have many unmet needs, and I think putting together everything is a big unmet need to really understand what is the contribution of each one, and how they interact with each other, and also the interaction with the environment, I think, is really important. That's why studies like what Kersey is doing at Rochester is so important to understand how the environment plays out, and that's really important. And I actually have a question. I was very intrigued with the facial AI. Do you think this is possible to incorporate, let's say, in a clinical trial in which, or not even a clinical trial, real life setting, when it's not perfect conditions, people take pictures with different cameras, different devices. Do you need to standardize it, or AI can kind of work with these limitations? What would you say?
Right. So for our study, we did choose to standardize lighting.
Mm-hmm. Yeah, it was a single center.
However, we found that the app itself regulates lighting conditions. Mm-hmm. So it won't take the picture unless proper lighting is indicated. Yeah.
That's great.
Because that's actually one of the things we have discussed about future studies, is incorporating in clinical trials and allowing the patients to take the iPads home and do daily updates of that sort.
Mm-hmm.
Yep.
Exactly.
And then if I could actually circle back to the unmet needs, I think it's of great interest, and we've had many remarkable speakers today talk on pathogenesis and then key markers. However, I think it's something as simple as future studies on adherence and mm-hmm. what factors are impacting adherence in specific patient populations. I think that's something that we should also continue looking forward in the future.
Definitely, for AD and beyond AD.
Correct.
And is that app widely available?
It is not. It is not as of now. We're hoping that the studies that we are conducting with the company will allow it to be more readily available, not just to clinicians, but to consumer use in the future.
That's very cool. Yeah.
My question is probably more towards Dr. Yuskowicz. When you talk about the neural inflammation in AD and then the connection or brain activation, with scratch, itchy, and then the brain. So you mentioned that there is the imaging technique showing the hypoinnervation with healthy skin and AD skin patients. So kind of a dilemma to me is if there's a less nerve system, sensory neurons in the skins, how can they sense either a scratch or any kind of signals from the beginning, and then whether they're like other first responders, so they can or conduct or signaling between the different entities. And the second one is kind of following that. We know that. Or with my limited research, I mean, from the cosmetic field. So when we look at the younger and the older patients, we know that there's also loss of the nerve density on the skin. So does it mean that these people are less sensitive to all this kind of stimulus and stuff like that? Thank you.
No, it's excellent comment. So my answer to you is it's not about quantity, it's about the quality, what they convey. So the amount of nerve fibers that transmit itch in comparison to all the nerve fibers is very limited. But as I mentioned, I think we know from the Chinese culture, yin and yang, there's a kind of also good nerve fibers. By the way, I mean, there are C nerve fibers that transmit pleasure, and scratch goes with pleasure. So we need to understand better what are the good nerve fibers and the bad ones that transmit itch. And clearly, as I mentioned, even in the brain, as you could see, there was less gray matter in patients with atopic eczema than when they improved. So there's some kind of signaling there that goes from brain, skin, and vice versa. So the message is really what nerves convey, and it's not numbers. And there was someone asked me in the audience before, when you do mice studies, that's a big difference from humans. When you induce mice atopic eczema, it's a cute type of model, and there's more nerve fibers when there is itch. And it's totally different in humans. So it's clearly, and it's not just unique to atopic eczema, the majority of chronic itch patients have less nerve fibers in the epidermis.
I just had a quick extension to that question. What about the non-lesional skin? So do you see that same decrease in non-lesional skin?
So I showed in that image, and I didn't go into it, there is less nerve fibers, and it kind of coincides with Emma's work that showed that a healthy, soaked-looking skin in atopic is not really healthy. And we have to look at atopic eczema as a systemic disease in a way. So epidermis, dermis nerve, there are a lot of components there. So it's not surprising, but clearly the number of nerve fibers were less in the lesional versus the non-lesional, but not like healthy. Does that answer?
Yeah.
Thank you all for the great education today. I work more on the CTCL side of things, so this was very helpful and just kind of, I'm curious in your center's experiences, just in general in this area of like biologics, have you seen certain acceleration of disease? Like we've had these patients come in where it's been misdiagnosed as eczema for several years, turns out it's some kind of, Sézary or low-grade MF, and then on certain biological inhibitors, they've progressed or had kind of explosion of the disease. Have you had experienced that with specific inhibitors versus others? We've kind of seen that more with these TNFs and other autoimmune conditions. And then there was this large controversy about dupilumab versus if that's treating or if that's kind of exacerbating, and just kind of speaking about this overlap of how biologics can maybe accelerate disease versus help in eczema, certainly.
Maybe I'm not clear. What did you say? The dupilumab may exacerbate AD?
No, exacerbate in lymphomas, so cutaneous lymphomas. Yes,
I see.
Which I think is more a question for you.
Yeah, that's a big.
Now it makes more sense.
Yes.
Okay.
Yeah, so it's a big controversy, right? Whether dupilumab, it's a big controversy whether dupilumab improves, has no effect, or makes eczema worse. I think, mycosis fungoides worse. It's difficult to say. Many patients that come in with mycosis fungoides exacerbation under dupilumab, they have, of course, initially been misdiagnosed, I think, as eczema. I think they had MF right from the beginning in most patients. There are other patients where you clearly have childhood atopic dermatitis and then they have superimposed MF. Just had a patient last week that got treated with a JAK inhibitor. And sort of there were different kinetics. The eczema responded better than the individual MF lesions. It's very individual. I think that in most cases dupilumab has no effect on MF. I think it just doesn't work in MF. Many patients that have then the worsening under dupilumab, it can, it could also be just the natural course of the disease and it's an unmasking. But it remains a controversy. I personally believe that dupilumab has little effect on MF.
I want to say something good about dupilumab. I agree fully with it's not a good treatment for MF and maybe can exacerbate in some cases. But in other cancers, it's important to know that actually dupilumab has a protective effect. I'm not trying to tell you to start using it for cancers of different types. I think we're the first to actually use it in patients who have advanced melanoma. Emma used it in multiple myeloma with excellent results. And I have to tell you that actually our colleagues in the cancer, they didn't get an FDA approval, but I just had last week, was a patient on a PD-1 inhibitor that had horrible flare of his atopic eczema, and the colleagues in the cancer know that to give dupilumab, that it saved this guy's treatment because the PD-1 work, when they exacerbate the eczema, they really flare the eczema, but they're life-saving. And this patient totally cleared with his eczema and could proceed with a PD-1. So I want that message to be conveyed because I know that the company is considering maybe getting in a label for that, but in reality, this is a lifesaver for some of the patients.
This is such an important point that you make, and actually our work on multiple myeloma showing that, and it happened serendipitously at Sinai. So what happened is I started to have some referrals from people treating multiple myeloma due to the patients having AD. And we treated with dupilumab with their permission, and we showed in 3 patients that dupilumab actually not only, of course, improved their AD, but these were the patients that they could stop their chemo and other treatments, and that is leading now to trials with dupilumab as potentially an adjuvant done at Sinai by Miriam Merad and her group in lung disease and other cancers. And also I want to point out that Nick Gulati from Mount Sinai also published a paper showing that in dupilumab-treated patients, they are numerically and some significantly decreased cancers. Not all cancers, all cancer types, but some cancers are decreased. So this is very important, and I think it's an important message because, you know, sometimes patients and physicians are like, oh, we don't want to give biologics, it may increase cancers. That's not happening. And we are nearing the end, but I think one additional question we can still take if anyone has a burning question. And if not, I want to thank all of these presenters, amazing presentations, and thank you for being here in this very cold room. I was shivering the entire time. Thank you so much.